Hello guys, I hope you guys are doing fine. And today we'll be talking about human fertilization, the various activities that lead to human fertilization, and how this human fertilization is able to come into play. Okay, now the human fertilization is a necessary process for what the production of embryo. Of course, we all know that. Results in the fusion of the what male and female pronuclei, all right, to produce a cell which is what diploid and chromosomal complement. Okay, this paves way for what embryogenesis. Now, fertilization in the humans take place in the ampullary region of the fallopian tube. The ascent of the spermatozoa to this part of the tube takes between 2 to 7 hours after deposition in the female genital tract. Okay, now this is the coronal radiator. This is just trying to show us the layers of the matured ovum. Okay, now you see once the sperm comes in, you see all these mechanisms that will be set up to stop polyspermy. Okay, that's fertilization by two sperms. Let's move. We'll understand this better as we talk. Now it is accomplished by what swimming motion of the spermatozoon and uterine and rhythmic contractions and fluid current in the female reproductive tract. That's the movement of the sperm to the ampullary region, okay, of the fallopian tube. Now, of the total number of the spermatozoa deposited in the vagina, only 1% reaches the cervix. Fertilization of the ovum requires that the spermatozoa undertakes certain activities, okay? Right now, first of these activities was capacitation. Now, capacitation is a process of spermatozoon activity, activation to achieve what full motile and fertility capabilities. Capacitation takes place within what 10 hours of ejaculation. It is a necessary step preceding the acrosome reaction, without which fertilization is impossible. Okay. Spermatozoa are inactive when expelled in ejaculate, okay, and they can't fertilize. Okay. Now there are some activities that are meant to take place to activate this spermatozoa so that they can be able to fertilize an ovum. Now, but with contact of semen fluid, okay, with contact of semen with fluids of the female genital tracts, multiple changes occur that activate the sperm for fertilization. Okay, possible events are what washing off and dilution of inhibitor factors, the cholesterol molecules that are constantly released into the fluid, into the vas difference, which adds to the membrane covering of the sperm cells, especially the acrosome, are washed off as the sperm cell swims away from this fluid towards the uterine tubes. Okay, cholesterol molecules are washed off. There's increased permeability towards calcium by the sperm cell membrane, leading towards a more forceful contraction of the flagellum, okay, making the movement much more propulsive. Now, membrane depolarization enhancing what exocytosis or the acrosomal enzymes. Okay. Now, how about we just spoke about capacitation, where everything covering the sperm head is being washed off. Okay. Now let's talk about the acrosomal reaction. This reaction permits the penetration of the ovum by the spermatozoon. Okay, the spermatozoon is attracted to the binding receptor of the ovum by the release of specific chemicals by what chemotaxis. Okay, all right. When the ovum has this specific chemical that is attracting the sperm. Okay, that's attraction tools through chemotaxis. Now this is just a scheme of what acrosomal reaction, okay, where the enzymes from the acrosome are released, okay. You see, these enzymes are help in the boring, okay, of um, of the ovum, okay, for fertilization to occur. Now, also glycoproteins on the outer surface of the sperm then bind with the glycoprotein on the zona pellucida of the ovum, okay. Um, Contact between the ovum and the spermatozoon causes the breakdown of what? Acrosomal layer. This release what? Hyaluronidase and trypsin like acrosin and zonal lysine and fertilin. There is also penetration of the zona of the coronal radiator by the release of what? Hyaluronidase from the acrosome to digest what? Cumulus cells surrounding the oocyte and exposing acrosin attached to the inner membrane of the sperm. Okay, this hyaluronidase will what? Digest the cumulus cells and help the sperm to move into the ovum. Within minutes, these enzymes open a penetrating pathway through the zona pellucida to the inside of the ovum. And this is aided by what? Constant propulsive force from the sperm flagellating cell and tail. Okay, the sperm move in a corkscrew manner. Okay, helping it bore deep. Okay, with these enzymes still acting and the sperm pushing, they are able to bore deep into the ovum. Now, binding occurs at the what? Posterior region of the sperm head. Now, within another 30 minutes, the sperm membranes, the cell membranes of the sperm head and of the ovum fuse with each other to form a single cell, which is a diploid zygote, okay, which will undergo mitotic divisions to become the embryo. The second meiotic division is then what? Activated to completion by the protein phospholipase C zeta. Okay. All right. Now, this is still another. I would say this is still um, the cortical reaction or the zonal reaction, uh, acrosomal reaction, where the sperm coming in contact with the ovum degenerate the what? Acrosomal layer, releasing the acrosomal enzymes such as the hyaluronidase, the trypsin like acrosin, the lysine, and so on. Now we say that the two haploid nuclei, that's the paternal and the maternal, fuse to form a zygote. Okay, and also cortical reactions ensues what? Immediately after fertilization, preventing polyspermy and possibility of what? Triploidy. This is characterized by massive exocytosis of cortical granules. This contains a mixture of enzymes, including several proteases. Now, this is seen shortly afterwards, sperm oocyte fusion. Okay. Now, zonal pellucidal structure is what? Altered. That's zonal reaction. And specifically, zonal reaction includes what? The hardening of the zonal pellucida. This is analogous to the setting of what? Setting of a concrete. Now, this is a runner-up spermatozoa that have not finished traversing the zonal pellucida by the time of the hardening occurred are stopped in their tracks. Okay, and also destruction of the spermatozoon receptors in the zonal pellucida, therefore, any sperm that have not yet bound to the zonal pellucida will no longer be able to bind, let alone fertilize the egg. All right. Now, zonal reaction prevents what? Polyspermy in and mammals. Okay. So this is how outermost layer of the ovum looks like, and this is the spermatozoa, very spermatozoa, and this spermatozoa having this light on his head looks like the closest to the egg, so it is like the chosen spermatozoa. He won the race. Okay. Now, following fusion, the sperm head is incorporated into the egg cytoplasm. Sorry. All right. I think I made. Uh, Following fusion, the sperm head is incorporated into the egg cytoplasm. The nuclear envelope of the sperm disperses, and the chromatin rapidly loosens from its tightly packed state into what? A process called the chromatin loses from its tightly packed state in a process called what? Decondensation. Other sperm components like mitochondria are degraded rather than incorporated into the embryo. and two genomes condense into the chromosomes, thereby reconstituting a diploid. Okay, so this is just a schema for acrosomal reaction. Right. With that, guys, we are done with human fertilization. Okay, so I'll see us very soon, and bye for now, guys.